Stevens. Yeah, so Stevens can take data as well as so we can click like really.
Okay, so I'm gonna do a quick attendance. Oliver,
Here's his next question, like what effect does that have?
Francisco, Genesis, no. Emmanuel, Deandra, Ian, sí. Luis Landrau, Fabiola, sí. Patricia Nayet, Sebastián. How will she get moved out of here? Okay, so good morning, everyone. I'm going to go ahead and get started with a brief introduction of our guest speaker today. So today we have the honor of having Dr. Tommaso Cupido. And as far as his educational training, he received his Bachelor of Science at the University of Milano, Bicocca in Italy, his PhD at the University of Barcelona in Spain, and went on to do a postdoctoral work at the Rockefeller University located in New York. Currently, he is an assistant member of the St. Jude faculty. His research interests consist of mechanism and regulation of protein machines, chemical probe discovery, and with that, also diving into cancer biology. So I will turn over to our guest lecturer. Your mic is off, sorry.
All right. Hi, everyone. Thank you, Arthur, for this very kind introduction. It's a pleasure to be here today with you guys. We're going to talk a little bit. Hi, hello. I can see you. All right. Wonderful. So I can see you have a full house. So how do I share the presentation?
You should be, let me see, if I haven't, probably need to.
Okay, I think we can do it like this. Okay.
Yeah, I should be able to, yeah.
Do you see my screen?
Yes, we see your screen. Mm-hmm.
Do you see the presentation or you just see the whole screen?
I see the presentation. It's not in like slideshow mode, but I see it.
What about now?
Yeah, now we can see it.
Okay.
We can see the individual small slides. I don't know. Let me see if I can. I think it's fine. We can, yeah.
I have two screens. Probably that's part of the problem.
That's okay. I mean,
I can disconnect one. The second, it resets. Okay, so what about now?
it's not in slideshow mode, but yeah, now it's perfect. Thank you.
Wonderful. All right, so thank you for being here. And so for the next, I guess, 1 hour and a little bit, we are going to talk about chemical biology drug discovery. That's what we do here at St. Jude in the Department of Chemical Biology and Therapeutics at St. Jude Children's Research Hospital. So how many of you have an interest or know about drug discovery or chemical biology?
Seems like half the course does.
Half the course does. That's perfect. Yeah. Okay. Okay. So guys, please interrupt me whenever you want. Right? I'm gonna time to time sort of stop and try to engage with you to see if some of the key points are taken home, have been taken home. Okay. All right. So what my lab does, and what has been the focus on my research for forever, has been the design and the development design of small molecules. Okay, the rational development of small molecules. Okay, so why do we like small molecules? Because they are very useful tools to understand biology, to examine and understand biology. Okay, and not only of course we understand biology not only from a fundamental point of view, right, how things works in nature and in biology, but also because we can understand the biology, specific biology related to disease. Okay, so that connects to the other part, for which small molecules are very important, because they can be starting point for developing new therapeutics. Okay, because there's no good way to test a therapeutic hypothesis other than to use a therapeutically useful tool. Because you can use genetics to understand the function of a protein or the connection of a protein or a macromolecule to a specific biological phenotype, a specific disease phenotype, right? But the only way that you can test therapeutic hypothesis if you have a therapeutic useful tool. Not all therapeutically useful tools are small molecule, but the vast majority of them are. Okay, so we use small molecule to both try to understand biology and diseases and to develop new therapeutics. Obviously, a small molecule per se, once we discover it, and even when we tweaked it, is not a drug. A drug is something much more complex. We're sort of going to touch on that a little bit, right? Drugs needs to be efficacious and safe in a human, or in an animal, whereas a small molecule, as we intended, doesn't not have those requirements, as long as we can use it to learn something about biology. Okay, so what is special about these small molecules? The small molecules is that they are just few hundreds Daltons in size. Okay, and what we ask these small molecules is to go and find the target, right? And the target biomolecule is typical biomolecule, 99% of the time. It is much, much bigger, right? So we are asking these very few atoms that are assembled rationally to modify the behavior of a target macromolecule, which is much more complex and much bigger than a small molecule. Okay. Now, small molecules, I talked about rational design, but we didn't invent small molecules, right? So nature or biology uses small molecules all the time. It uses as a signaling molecules most of the times, right? And the reason why they're useful for signaling molecules is that they diffuse very fast compared to biomolecules, right? They can diffuse inside the cells, reach any protein in the cells in seconds, typically, right? Or they can use different tissues in the body just by systemic administration, right? Nature uses it as chemical warfare. Antibiotics is a classic example, right? That regulates competition between the different organisms in nature. Poisons, and nature makes some of the most powerful poisons that we know. Okay, and they're also used in biology as cofactors for enzyme. We use the property that is the reactivity of small molecules. They're like catalysts, right? They're not really consumed during the process, but they're just used by enzyme to do some useful chemistry on other biomolecules. And they're also used as nutrients. I don't want to talk about that because there's destroy to get energy from it. And they're also used as energy currency. Okay. So nature has learned how to use small molecules as building blocks and energy currency and as signaling molecules for doing a lot of useful things. Okay, so when we rationalize how to design and develop small molecules, we try to learn from these principles that nature has developed across billions of years of evolution. All right, so regarding to signaling, using small molecule to signal, right? So as I told you, small molecules are few hundred Daltons. They bind to a pocket in the protein. They get desolvated as they bind to a pocket, and they bind to this little pockets in these large macromolecules. And you have to imagine like macromolecules as a dynamic entity, right? A macromolecule is alive, okay? Small molecule is not alive, small molecule is dead, okay? But the protein just sort of travels towards, explores different states during its lifetime, right? And each state can have different functions, okay? Each state is a 3D structure. You know, the structure relates to function. So each different 3D structure may have a different function in sense, right? So this is depicted in this scheme here. And so you have an exemplary target micromolecule, a protein, let's simplify it. And this got 3 states, which is in dynamic equilibrium. Each of these states may have a specific function. And the function, once you have one, you sort of inherently inhibit the other function, right? Because if you can imagine a distribution of this protein across 3 different states, so like a third of the protein does each of the functions. Once this equilibrium is perturbed, you only have one, so the other 2 functions are inhibited, right? So, and this is how small molecule can act as signaling, right? So they bind to this pocket and they sort of perturb this equilibrium. Do you see my cursor, guys?
Yes.
Yeah.
Yes.
Thank you. Okay, never mind. I'll just use the cursor. Okay, so this is depicted here. The small molecule, this small molecule cannot bind to the state of the protein A, cannot bind to C, but it does bind to B, right? This perturbs the equilibrium of the states of the protein, so that only one of these states is stabilized by the binding of the small molecule, and only function B becomes predominant with inhibition of function A, function C, okay? So this is the same principle that we apply when we design small molecule in order to sort of modulate protein functions toward the function that we want. Or sometimes you just want to shut out every function of the protein, right? That's sort of complicated to do, but most of the time, the small molecule stabilizes one state, and that one state carries one particular biology with it and shuts down the other parts, okay? Move on. Okay, it looks like I did something and now I cannot move on. All right. So this is the challenge, this central challenge in chemical biology, okay, that we have. So we are asking this small molecule, few daltons, to enter into an extremely complicated system, which is a cell, right? And to find its target macromolecule and to modulate it, okay? And you can see how this is such a difficult task because you can understand how complicated is the environment, how many other proteins and targets macromolecules there are in a cells. And when we talk about the drug, then we go to the ultimate complicated system, which is the human body, right? So we are asking this small molecule to go inside the human body, find its target modulated, be safe, right? And ameliorate a certain disease phenotype. This is an extremely challenging task, okay? All right, so there's a key concept when we start to design small molecule. It is, we want the molecule to be effective, and we want the molecule to be selective, okay? Otherwise, it's not useful for understanding biology because we don't know what we are perturbing, okay? So we're gonna go a little deeper on these 2 concepts of effectiveness of a small molecule and selectivity, okay? So just a tiny little bit of physical principles here because they are important. Okay, this is how we rationalize. I think most of you are familiar with this concept that I'm gonna try to summarize here very quickly. Right? Sorry. And so we describe effectiveness as how much of the target of the protein we have bound to the small molecule. Right? This depends on the free energy difference between the free protein and the protein bound plus the small molecule, right? And we sort of characterize this as a chemical reactions in which you have A and B. This parenthesis stands for the free concentration of the 2, and then you have a forward reaction in which you have an association of the 2, and a reverse reaction in which you have a dissociation of the 2, right? and you go from unbound to bound. So the ratio between the rate constant of the association and dissociation is called the dissociation constant, we call it. And we should be inverted. I'm sorry, there's a typo here. It should be A versus A times B divided by AB, because the dissociation constant, this looks like the association constant. Okay, so the ratio between the two Ks is what we call the KD. And the KD tells you about how much energy there is in the binding of small molecule, right? So that's the sort of the physical concept. But it also tells you something different, okay? So this graph over here depicts the fraction of the protein bound versus the compound concentration. It follows the typical hyperbole, the absorption law that regulates gas absorption into a surface, for example, right? And this is a universal law. And what you see is that we have, so as we increase the compound concentration or the small molecule concentration, we increase the fraction of the target bound, right? So, and the relationship is asymptotic, okay? What it means that we can occupy the first molecule, the first target biomolecule very quickly, but then as we go higher and higher in concentration, it takes more and more of the ligand or of the small molecules to finish complete binding of the target protein, okay? So this depicts a situation in which you have a dissociation constant of 200 nanomolar of 0.2 micromolar for an expected target. So the first point that I'm going to make is that to be effective, the small molecule must be potent, okay? The KD describes the concentration in which we have 50% occupancy. So the ratio between bound and unbound is 50%, right? And the lower is the KD, the less molecules we need in order to occupy the target, okay? But I want to be reminded that the relationship is asymptotic, okay? So let's go a tiny little bit deeper on this, right? This is a classic view of a protein plus a ligand, right? I talked about this already in the previous slides. There is an association and a dissociation. You have a protein plus a ligand, it becomes a protein ligand complex, okay? So, and this is sort of the same concept, a little more scholarly depicted, right? So you have at zero compound, you have zero protein bound. As you go up in ligand concentration, you have the KD, in which you have the fraction of 0.5 bound, and then the asymptotic relationship. And it takes a little bit, in order to occupy completely, so the fraction 1.0, well, you need a lot of small molecules, okay? So this is depicted clearly here, right? So the very, very low ligand concentration, the fraction occupied is almost zero, right? You can still have small molecule around, but the potency is not enough at that concentration, at that very low concentration, to occupy any protein. Okay, when you increase the ligand concentration, we have in this situation, when the concentration of the ligand is equal to the KD, you have half occupied, half non-occupied, right? And then we have to go very, very high in concentration, about 100 times higher, in order to get higher than the KD, in order to get a full occupancy of the target, right? So we use this simple law to precisely quantify how much free and bound protein we have. So how much we have of function B or how much we have of function A, okay? Now, this segues into this different concept. It is ancillary concept, which is the concept of specificity, okay? Now, let's imagine that I have two biomolecules, right? Two proteins, they're kind of similar among them, but they have vastly different dissociation constant between the two, okay? So for one protein that is my desired target, what we call the protein that I intend to modulate with protein, with the ligand, the KD is 10 to the 9 molar, which is 1 nanomole, okay? But then I have another protein, which KD is much higher, and is 10 to the 5, 10 to the minus 5 molar, which means 10 micromolar, okay? So there is a huge 4 order of magnitude difference in the KD, okay, between these two ligands, which is these two targets for the same ligand, okay? So now, if I tell you that give this ligand to the system in a concentration of 0.1 micromolar, which is 10 to the minus 7, which is the half point between 10 to the minus 5 and 10 to the minus 9, then we are in a situation in which we have a drug concentration that is 100-fold higher than the KD for the intended desired target, which is this orange guy here, and is 100-fold lower than the KD for the non-desired target. Okay, so in the situation that we have is depicted in this square diagram here. So you only have your desired target occupied, and essentially you have none of the undesired target occupied because the difference between the KD is so huge. Okay. Now the message is the following: is that the small molecules needs to be potent for on target. I mean the KD needs to be very low, right? And the KD for the unintended target needs to be significantly higher. Now the difference of 4 orders of magnitude is sort of ideal. We typically cannot get that. Okay, because the proteins, some proteins are very similar among them. We don't have enough structural feature to differentiate between them. There are some tricks that we can play. Okay, but this is sort of what do we think about when we go into designing small molecules that can target a specific protein, for example, a disease-related protein. Okay, so that's the situation depicted here. So 2 targets. The blue line is our intended target, and the red line is the non-intended target, right? So here the KD difference is only 10 times, right? It's not 10,000 times as before. We only have 10 times difference in the KD between the intended and the unintended. Do we have any selectivity here? So you can see that any concentration of the drug here, as I increase on the y-axis, the drugs of the compound, right? The x-axis is the concentration of the compounds. Essentially, at any concentration of the, sorry? I thought I heard.
Sorry.
Okay, never mind. So at any concentration of the compound, we essentially have both targets occupied to a certain extent, right? So we don't have selectivity. So we have to sort of deal with that. We hope that the target A1 is not, that hitting both targets is not detrimental to what we want to study, or is not detrimental for curing a disease, right? That it's only one of them is important. We can target both, but only one of them is important. Now, of course, we have the one that has the lower KD. There's more occupancy. The one that has the one that has the lower KD has more occupancy, and the one that has the higher KD has lower occupancy. So there is a difference, but both of them are hidden, right? If the KD difference is not high enough. All right. So enough with the physical principles. I hope that this message is sort of clear. Now, we deal typically in small molecules with 2 vastly different concepts, right? So one of them are small molecule probes, and the other one is small molecule drugs, okay? The initial stages of the process of the discovery and development is kind of very similar, and there are parts that are identical. But when we deal about drugs, we have a different endpoint, right? So we need to perform animal testings, we need to go over clinical trials, and finally we need to go to the regulatory review of the process, which ends up being approved or non-approved drug, right? And it also, the initial part of the process is kind of different, right? So we study the disease, we study the biology of the disease, and we came up with a target, with one biomolecule or one process that is implicated in the biology of the disease. So we came up with an hypothesis, a therapeutic hypothesis, in which untested, in which we decided that modulating that process of that molecule can ameliorate a disease phenotype, okay? And that's not the same when we talk about small molecule probes. In the small molecule probes, we don't necessarily link a process, a biological process, to a disease. It's enough that the process is interest, or is there something unknown about the process for us to want to go in and develop a small molecule that targets that process, okay? Process or biomolecules.
Okay, we have a question in the class. Just
Sure, please. I can't.
Hi, can you hear me?
Yes, now I can hear you.
Uh, Hello.
Now-
Yeah. There you go. I had a quick question based on what you were presenting on the KD, and it sort of correlates with talking about compound discovery.
Yeah.
Let's say I'm a synthetic chemist, and let's say I have a compound that seems to have promising, it could become a bioactive molecule, let's say for cancer, for example.
Mhm.
Do I have, do I have to do a study for the KD, for it to have a low value for me to argument that this could be a potential drug, or is that something that is done way after?
Okay. Look, in order for you to develop that molecule, right, to make the molecule that has bioactivity in the first place, most of the time you already have a KD measurement. Okay? Most of the time. Now, it's not necessary, right? If you take a plant, you chop it out, and you have an extract, and you show that the extract has an activity, you don't need to determine a KD, right? But for you to get to the full development of the molecule, most likely you need to have some measurement of the strength of interaction to the intended target, at least in modern drug discovery. That's actually a very good question, because before that was not considered that important. Okay, before there's some bioactive small molecules, it codes something, I throw it to the cells, it codes something, I go to animal. If it's active in a disease model, I'll give it to humans. We don't do things like that anymore. All right, that has caused tremendous successes. That's like, from the 50s to the 90s, that's drug discovery based on natural products, mostly, right? Taxol, mainstay of anti-cancer therapy, breast cancer came from that before it even was discovered that it targets tubulin, and the affinity for tubulin was measured. But it is unlikely that any drug discovery program today does not have a target defined. Once you define the target, you do define the affinity of the KD for that target, because that provides a measure for you to make the compound better and better and better and better, right? Otherwise, you're blind. Does it make sense?
Yes, thank you.
Yes. Any further questions? All right. So let's move on. So where were we? So the small molecule probes and the drugs, right? So you can see that the end point of a small molecule probe is different from the one of the drugs. So the small molecule probe, you get just new insight into biology. And if you're lucky enough, you can get that molecule and take that molecule and enter a drug development program right from that molecule, okay? But the output is different. Okay, it's a molecule that is useful to understand biology in the case of small molecule probes. Now, there's another difference. So whereas in drugs, we need to do animal testing, right, to set up the dose, to provide proofs of efficacy, proof of concept, small molecule probes, is fundamental that we assess selectivity, okay? Because we need to understand how many targets the small molecule has. It is extremely difficult to do that. Okay, so we typically settle for something in the middle. All right, so that's another difference. Drugs don't necessarily need to be selective, they just need to work. Okay, small molecule probes need to be selective, otherwise we do not understand, or at least we have to know the difference in KD between the different targets so that we can sort of rationalize what we see when we use a small molecule. Now we talk about the difference, and there is also the middle part is actually the same for both drugs and small molecules. There's always a compound discovery phase, and there is a compound optimization phase, always. Right? The small molecule you discover in the first place, doesn't matter the source, even natural from chemical collections or from your own brain, you always have to take medicinal chemistry and optimize the molecule as much as you can. All right. Now, where these compounds come from? How do we find them? Okay. So these are four kind of strategy that we can have, right? Typically used. So the first strategy we want to talk about is we just observe what nature does. Okay. We just take the molecule from nature and then we tweak it. Well, all the idea of how to modulate the target, it comes from nature. We don't even understand it, right? Fully, most of the time, at the beginning at least. Another strategy is that we copy what nature does, okay? We don't necessarily take the molecule that can work from nature. We just study how small molecule and protein interact in nature or in biology, and we try to copy some aspect of those. Okay, there's an element of rational design there. All right, and then we have other strategies that are collectively called de novo. Okay, in which we just test large collections of chemicals that have drug likes. Okay, and we test it in typically two ways, either in biochemical screenings or in phenotypic screenings. Okay, so we're going to talk first about those de novo strategy, and then we're going to talk about the other ones. Now, most of the time, modern drug discovery program or modern compound discovery program use combination of all these strategies. There's not a single strategy that can give you success, right? Typically, limited with resources, right? But we try to explore multiple strategies. In the science literature, different labs, different people explore different strategies, right? More shots on goal. In big pharmas, also multiple strategies are explored at the same time until one that looks more better is found, and then all resources are dedicated to that. All right, so let's talk about high throughput. The first part is biochemical screening, right? We call biochemical screening high throughput. It means we can test. So since we have, frankly, our understanding, despite decades of knowledge, our understanding of how to design small molecules, it is still very limited because we do not fully understand the energetics involved with binding a small molecule to a protein, modulating its conformational landscape. Each conformation is a different energy. So how the conformational landscape is modified by binding a small molecule to one, right? It is frankly very complicated and we don't fully understand. So most of the time, what do we resort to is just blind testing, okay? So we assemble this large collection of drug-like chemicals that have properties that are suitable for them being developed and modified, right? And we test them in a test tube, okay? Now, since the advent of recombinant DNA biology, the recombinant DNAs, which is like 30 years ago, now we can sort of produce recombinantly pretty much any biomolecule of interest. Okay, it's not exactly true, but we have robust, let's put it this way, we have robust methods to express and purify defined materials, defined biomolecule, right, in a test tube. So this is the process of handling all these molecules is these two pictures here on the left. This involves typically some robotics because there are hundreds of thousands of millions of compounds, right? We have that here at St. Jude, an entire space dedicated to the robotic testing of 100 tons of more molecules. Okay, so once we have that hardware, where infrastructure.
Also, Tommaso, sorry. Just to clarify, they will be learning about HTS later in the semester, so you also don't. John is gonna give them a very much in-depth section on HTS. So you also don't have to go too in-depth if you prefer to focus on other areas as well.
All right, great. So I'm just gonna blast through this. Okay. So essentially, this is biochemical screening. You need a target, a protein. This is the purification of a target protein. You see, you extract it from a cell. It's very dirty. You go through different purification proteins. When you have purified material, then you're ready to go. You can design your assay. Okay. Design your assays in vitro with purified biochemical material in a defined system. If the protein is an enzyme, you can sort of design colorimetric assays, typically of fluorescence-based assays, based on the reaction that the enzyme does. Okay? After you have the assay in place, you do assay miniaturization, and you have an output. The output is the 1 out of thousands of these compounds is able to modify that reaction. Okay? Another another branch of screening is phenotypic. Okay? Again, there's infrastructure, is typically microscope involved, and there's compounds are already in a place, as you can see it here, and there is some sort of signal that you get from the cells. Okay? And in this case, the main difference, so all the green dots are small molecules, each well is one molecule. All the green dots are small molecules that sort of modify whatever parameter you're looking for in fluorescence microscopy. Okay? And this is called phenotypic because we don't know, this is different from a reconstituted system, right? So we don't know what the molecule is doing. We just know that it's modulating the signal that we want. That's the main difference, okay? All right, so there are some very advanced phenotypic screening strategies here, right? That I show you that was just one marker, right? The green signal in the cells, but we can add multiple markers in a technique called cell paintings, right? And I image them all at the time, right? So typically this is coupled with some artificial intelligence, machine learning methods to analyze these images to understand what is happening to the cells, right? So the difference between what I showed you before is that now we have some idea that that compound is doing something specific to the cell, okay? And we can cluster all the green dots that we have, all the compounds, according to some features that machine learning tells us. So this cluster of compound does all the same things. This other cluster of compounds does all a different thing. Okay, all related with the disease biology that you want to study. Because there's many ways that you can sort of target disease biology. Biology is complicated, right? There's not a single target that determines what biology is involved in a disease. Okay, now this advanced technique are used nowadays to sort of, they're very useful, and although they are blind, and they're used to sort of tackle some most challenging cases in drug discovery, where, for example, the modulation of biomolecular condensate. I don't know how many of you have ever heard about biomolecular condensate, but I'll just summarize it in a certain way, simplistic way, so that these are just ways in which molecule interacts that are non-stoichiometric, okay? So they involve multiple low affinity interactions that are very highly dynamic, okay? And they cause this molecule to sort of coalesce or separate from the rest, okay? In a process that we call phase separation. And so these clusters of molecules behave different from the molecule in solution. They exchange molecule with the solution, but once they are in the condensate, they behave different. So they can exert some specific biology, okay? So one of these proteins that can form condensate physiologically or non-physiologically. So one of the proteins that forms condensate that we don't think is physiologically is a transcription factor that is called c-MYC. It's a very important growth-promoting transcription factor in cancers. And certain cancers have dysregulated this transcription factor. They have elevated levels. At elevated levels, higher to physiological levels, this protein is able to form this biomolecular condensate. So it is pathologically related. It's not physiologically. Right? So these imaging techniques are used to identify compounds that can modulate the assembly of these condensates in the cells. So why is this a particularly challenging case? Because as I told you, these are non-stoichiometry, right? All the things that I told you about, protein found in a pocket, a small molecule found in a pocket, having a KD, they don't apply here. We don't have pockets, right? Because the molecules are typically disordered in this condensate, right? So there requires new physical principle to be developed in order to understand how to modulate. But phenotypically, we can see that certain molecules do modulate, right? The formation of the function of this condensate. We don't understand how they do it, but it requires different physical principles. Okay, so we discussed de novo very briefly, and And now we're going to discuss the two cases in which one we observe the nature, how nature does it, and the other one in which we copy nature. Okay? There are any questions so far?
We're good.
We're good. Let's move on. Okay, I'm going to tell you this story. Okay? Sorry?
Oh, no, we're good.
Okay. All right, so I'm going to tell you this story, the story of this small molecule. The small molecule is called cyclopamine, right? This is the chemical structure. This is the space filling the picture. This is what the real small molecule, the electron, looks like. This is how we conceptualize it, right? So this is found in a plant. It's produced by a plant. It's called Veratrum californicum. And it was sort of, there was this phenomena in a Idaho farm in which in the 50s, so a lot of where these plants grows, in the prairies of Idaho, and a lot of like a sheep. I'm sorry if this is a little morbid here, but it's just trying to make a point. A lot of sheep were born cyclopic. Okay, so it means that the normal patterning of the face of the animal was broken. It didn't work, right? So they were failing to separate the symmetry of the face into two eyes and two ears, etc., etc. So there were some defects that prevented them to do that. So they found afterwards that the cause of this malformation were that these sheep were feeding on the Veratrum californicum. Okay, and so they were absorbing this small molecule that is called cyclopamine. So, and the question it becomes, how does cyclopamine, as an example, how we observe nature, how does cyclopamine causes this? What can we learn from it? And is it useful in any way for humans? Okay, this looks horrible, but can we use it for something? Okay. All right. So it turns out that on the 70s and the 80s and the 90s, people did many labs did a lot of forward genetic screens, right? Try to understand what regulates developmental of an embryo, the process by which one cells we get an entire organism, right? And with the specification of the various tissues. And they were doing that in a bunch of different model organisms, like the mouse or the Drosophila, the fruit fly, right? So this is simple forward genetics. You mutate, you look for the phenotype, something that's changed with respect to the normal, and then you try to understand what genes causes this. Okay? That's called forward genetics. Now, this you can see the normal embryo of a fruit fly, of a Drosophila, is this, but a mutant embryo, it looks like this. Okay? This is called the hedgehog phenotype. And then it so happens that the same genes were, when mutated, they cause this phenotype in the Drosophila. They were causing these phenotypes in the mouse embryo, right? So on the left, this is a mouse. On the left, we have a normal developing embryo of the mouse. But on the right, we have an abnormal developing embryo that is mutated on the same gene that caused this phenotype in the Drosophila. So both developmental related phenotypes. And if you look at this mouse developing embryo, as disturbing as it may be, it looks a lot similar to the cyclopic sheep of Idaho's found in the 50s. Okay? So that was the idea, right? Cyclopamine must target some proteins in the must target this pathway controlled by this gene, which is called hedgehog. Okay? So then follows decades of molecular biology, structural biology, and genetics work. We now have a near complete picture of how the pathway looks like in the developing embryo. And by the way, this pathway is conserved from the Drosophila to human. So this pathway regulates patterning of the embryonic development throughout higher eukaryotes. Okay, and the pathway works as such. So hedgehog is a secreted protein. It binds to patch, which is a transmembrane repressor, a transmembrane protein, and inhibits it. Okay. Now you can see this is the double arrow. This arrow means inhibition of patch. Hedgehog inhibits patch. Patch inhibits smoothness. So which means there's a double negative here, right? That once I inhibit patch, the result is I activate smoothness. Smoke is called smoothness, right? So that's what it means. So if I give hedgehog to cells, patch is inhibited, relieving his inhibition on smoothness, which then this is an activated arrow onto the transcription factors again, which is called GLI from glioma, overexpressed in glioma, goes to the nucleus and transcribes genes that are needed for the developing embryo. Okay? So it turns out that cyclopamine, what it does, it binds to the transmembrane protein, smoothing it into a pocket. But the full understanding of the structural details of this interaction came only very recently, a couple of years ago. Okay, and you can see the small molecule cyclopamine bound to these transmembrane receptors, smoothing it. So by binding to smoothing it, it blocks its activation, right? So it blocks the ability of smoothing it to activate the transcription factor. So it cuts off completely patch and headshot. They don't matter anymore, right? And it directly binds to smoothing it and inhibits it, which prevents the activation of GLI. You don't transcribe genes that are needed for embryonic development, okay? Hence the cyclopic phenotype. So that's how it works, okay? So now, cyclopamine is not a good molecule for a drug, but that understanding prompt drug discovery program aimed at finding direct antagonists of smoothing it. I told you that this GLI is overexpressed in glioma, but also in other cancers. So this is a general growth promoting signaling cascade, and this is promoting growth not only in glioma but also in other type of cancers. So this widespread usage of the HL transcription signaling cascade by cancer prompted the development of the starting of the various compound development campaigns until Genentech, a company that is in San Francisco here, developed this small molecule called vismogenib. It looks nothing like cyclopamine, right? It's completely different structure, but it has the same basic mechanism of action. It binds to the transmembrane protein, smoothing it, inhibits it, and it blocks GLI signaling, GLI transcription activation. Okay, obviously you can't give this to kids that are developing, but in adult, the GLI signaling cascade does not have essential function anymore. Okay, only the developing embryo. So this is a safe compound and is now an approved drug for the treatment of basal cell carcinomas because it can be curable, right? It can cure disease, can cure the disease. So that's how these small molecule drugs came to be. All right. So if there are some questions here. Otherwise, we move to the next topic.
Yeah, no questions.
Wonderful. All right. So the next topic, how do we copy nature? This is also interesting, right? It's different from just getting the molecule from nature directly. We try to sort of understand how molecules and ligands interact in biology. Okay, so I keep cancer biology is sort of my bread and butter. So I keep referring to cases in which the discovery of this small molecule ended up being cancer drugs, but it's not necessarily needed. Okay. All right. So this is another very famous example, right? This is this picture that you see on the left is a staining of human chromosomes in a diseased patient that has chronic myeloid leukemia. Okay. It's chromosome 9 and chromosome 22. And you can see there is an abnormality, right? So there is the one on the left is the normal chromosome 9, and the one on the left here is the normal chromosome 22. Okay, but then we have two different sort of guys, right? If you have a 9 chromosome which is longer, this is just cells taking out the chromosome. You take out the chromosome from a dividing cell, you stain it with a dye, and then you put it on a microscope. That's what it looks like. Okay. So, and pathologists in the 70s were start taking blood cells from patients in chronic myeloid leukemia, and they were recurrently seeing this chromosomal abnormality in which a chromosome 9 is longer, one of the chromosome 9 is longer, and one of the chromosome 22 is shorter. Okay, so there has been a translocation, what we call a translocation in certain cells. Then the cells proliferate, and now this translocation is visible in a lot of the cells of the blood of that patient. Okay, so this fusion translocation, which calls a fusion chromosome, happens at particular two genes, right? It corresponds to the two genes, so it breaks two genes apart and it fuses them back. Okay, and it creates this chimeric protein, which is called BCR-ABL, because you got a piece of the BCR-ABL gene and a piece of the the ABL gene, and they form a protein, and the protein has a function, right? But the function is chimeric, right? It's not natural. So what is the function of this protein? The part that we are most interested in is the ABL part. The ABL is a protein kinase. Protein kinase means transfer is a phosphoryl transferases. So an enzyme that transfer the phosphate from the ATP, which is the donor, to a protein acceptor, right? And it does that catalytically. It does that multiple times. Okay. So by breaking the upper part, the most left part of ABL, we remove his regulatory part, and we attach another part from BCR, which completely destroys his regulation. So now the kinase is hyperactivated, okay? And it keeps phosphorylating uncontrolled. And it ends up that the protein phosphorylates numerous cancer proteins, cancer-promoting proteins, right? So once you have this in a blood cells, this will promote cancer growth, okay? So that's the understanding, biochemical understanding of what the protein does, what this kinase does. Now, hence the idea, right? If this protein binds a small molecule and through the binding of this small molecule, it transfers the phosphor group to cancer protein, maybe we can design a molecule that does the same, okay? That looks like ATP, it binds to the pocket like ATP does. ATP is a small molecule, it binds to the pocket, right? But in that case, we'll displace ATP, right? So the BCR-ABL will not be able anymore to phosphorylate cancer proteins, okay? So that's the basic idea that we sort of copy from nature. Okay, again, structure. Structure comes very helpful when you are very, very useful when you want to design rational drugs. So this is the structure of just the ABL part of the protein kinase bound to the ATP. Okay, there are two parts roughly. One is beta strand, the bottom part is the alpha helix fold. And ATP is bound in the middle. Okay, so, and these schematics on the right tells you a little bit what are the precise interaction that ATP does onto the pocket of BCR-ABL, right? So this is ATP, they got 3 phosphate, the third one is one that is transferred, is the ribose bridge in the middle, and then there is the adenine base. Actually, it's missing a nitrogen. There's a type, anyhow. So the adenine base, what it does, it forms 3 hydrogen bonds with the backbone atoms, with this part here of the protein. 3 hydrogen bonds with the adenine are the one that determines the affinity of the protein for the ATP. So this is the idea. We can take a small molecule that looks like ATP, and we can mimic those hydrogen bonding, okay, using a synthetic small molecules, okay? That will sort of mimic the energy that ATP does in binding, but will not outcompete it. So how do we outcompete it? So if you look closely at the structure, we see that the ATP is not a perfect fit. And that makes sense because ATP has to come in, phosphorylate, and it gets out. And then a new molecule of ATP, if it's too strong, it will never get out. Then there's no catalytic cycle, right? So we can identify by looking at the structure 2 small subpockets that are near the adenine, okay? So one we call it the exterior pocket, which is more polar because it's exposed to the solvent, and one in the interior, which is very much apolar, okay, because it's not exposed to the solvent. So the idea is we can copy mimicking this hydrogen bonding interaction of the ATP, but then we can add something more that occupies this hydrophobic pocket. It generates more interaction than ATP does. Okay, so this small molecule that is depicted here, R means pretty much anything, right? But this is the core of the small molecules. You can see that this phenyl group occupies the interior hydrophobic pocket. Okay, so now we have an energetic gain with respect to ATP. So now we can potentially, now you can outcompete ATP in the cellular interior, and this can be an inhibitor now, okay? So this process takes actually a long time, so right? And this is the original small molecule that was found by screening, biochemical screening of the ABL kinase set up a kinase reaction, you test a bunch of small molecules, you find one that looks like it can outcompete ATP. And this is the first one in the top that you can see here on the left, okay? Then you go through medicinal chemistry modification, right? So you try to add and modify groups in a way that you can gain more interaction and gain affinity, okay? So the binding affinity was actually very low on the first initial hit, right? But then you add this fluorinated phenol group here, and you start getting millimolar affinity, okay? And still not enough. Then various modifications. Then you add perhaps a pyridine group. A pyrimidine ring to occupy the other pocket, the front pocket, the polar pocket. Then we started getting more and more affinity, okay, for the compound. So this process is iterative. So you make modification, you test it, you measure binding affinity, and you select the molecules that have a stronger binding affinity, and you go on. And this process culminated to the discovery of imatinib, which is these molecules that we have here. This is essentially the same small molecule that was found initially, but it's been modified and enlarged to catch more interaction. Now, these small molecules apparently, this is the crystal structure over here of the imatinib bound to ABL, occupies the subpockets optimally, and it has nanomolar affinity. Right now, the KD of the small molecule is thousands of times higher than the KD for the ATP. Now we have something, right? Now we can outcompete ATP in cellular system, right? Indeed, this molecule is able to sort of kill chronic myeloid CML cells in a test tube, right? And is also effective in mice. Okay, so we can cure chronic myelogenous leukemia in a mouse using imatinib. All right, so this was a paradigm shift, but it's kind of the old story. It's about the 2000s this story came about, all right? But it was a paradigm shift in drug discovery, because before this, the way we were designing drugs was typically the way we described before, where we just test them and we use we were looking for most potent toxins, the things that can kill cells more potent, the most potent ways. Okay, so that's the mainstay of chemotherapies that works, but it is highly toxic to both cancer and normal cells. Okay, those are literally poison that destroys essential process for all cells. Okay, but the discovery of Gleevec exemplified that we can target a cancer-specific protein. That mutation is only in cancer cells, right? So we can target a cancer-specific protein with a process with a small molecule, all right? And therefore, that caused the shift towards trying to design rationally molecules that act on cancer-specific mutations, okay? And he got the cover of Time because it was so important in that way we think about drug discovery. Okay, how much time do we have left?
Oh, we have 20 more minutes.
Okay, great. Any questions so far?
Any questions? No, we're good.
You're good. All right. So that was great. Now we can start cure CML, and we had, and from then on we designed drugs in a different way. Okay, but is imatinib a selective compound? Does it target only ABL kinases? Right. So this graph on the left shows you the evolutionary relationship between all kinases in the human genome. It's a tree representation of all kinases. There are about 450. I don't know, 540. I don't know the precise number, but there are many kinases in the human genome, right? Some are, the closer they are in this tree, the closer it is their primary structure, right? The primary sequence, okay? The far apart they are in this tree, the more divergent is their primary sequence, okay? Now, you can see imatinib is a selective kinase inhibitor, not at all. Right? These three, the dots in these three, the size of the dots are proportional to the KD or the IC50 of the compound for that particular kinase, right? The bigger the dots, the stronger is the compound binding that kinase. And you can see there are, yes, ABL is probably the biggest dot, but there are many of them, right? So there are many other kinases that are targeted. And so drug is effective, but it's non-selective, okay? We don't know to which extent precisely this non-selectivity is important for its clinical efficacy, okay? But we'll discuss a little bit about that. Now, is that specific for imatinib? Well, if you compare many of the first generation kinase inhibitors that were developed in the first decade of the 2000s, they pretty much all suffer from the same disadvantage, right? They're all non-selective. Okay. Now we are getting a little better at selectivity, and but all these compounds went into humans, right? Most of them did not make drugs, probably because they're too non-selective. So they target many essential processes because they inhibited too many kinases. Okay. So this is the first kinase inhibitor ever found, staurosporin. It is like inhibiting all kinases. So why is that? Because all these molecules, they were designed to the same principle that they were designed, the Gleevec was designed, imatinib was designed. So they target the ATP binding site. Okay, so the ATP binding, this is the depiction of a protein kinases in a different way. And you can, the color, it's color coded according to conservation grade, right? So the ATP binding site is the most conserved structurally in all kinases, okay? And the strategy decided to target that pocket. So you can understand that it is difficult to achieve selectivity when you target a pocket that is highly conserved, okay? There are a lot of nuances to what I just told you. There are conformational changes that happens when the drug bind kinases, and those impact on the selectivity. But conceptually, when you target the active site of the enzyme, it is reasonable that is the most conserved part, right? Because nature wants to keep that throughout evolution. It diversifies other parts for functional diversification, but the core catalytic function of the protein kinase is the same across all kinases. So it makes sense that the ATP binding pocket is the most conserved site, okay? So there is a lot of interest now in small molecule development in what is called allosteric compounds. So allosteric compounds, by definition, are compounds that don't target the enzymatic pocket, right? But target alternative site of an enzyme, or they don't target the main functional site of a protein, but they target alternative sites. And the discovery of pockets that are allosteric is not a trivial task, okay? Because most of them are not visible in the state in which we normally see the protein. The structures can tell us, okay? All right. So now, selectivity of imatinib. So I tell you about what is this selectivity contribute to clinical efficacy or not for CML? Okay, so how can we approach that question? Okay, so I told you that it is effective against leukemias, or chronic myeloid leukemias, one subset of them that have BCR-ABL rearrangement. Okay, but when we started using this compound in the clinic, then we made an observation, right? The clinical benefits were typically short-lived, months. Okay, patients were doing better, a lot better initially, but then a fraction of them unfortunately relapsed. And once they relapse, the disease is not treatable, does not respond anymore to imatinib. It became resistant to imatinib. Okay? So the idea was, we'll take these tumors that are resistant and we sequence the BCR-ABL gene. Maybe something has changed, right? Cancers are genomically unstable. They keep mutating. That's what keeps giving them growing advantage in a challenging environment, which is the human body that is fighting against them, right? So they take the BCR-ABL gene from the cells and they sequence it. And the discovery was 30% of the time in relapsed patients, when we sequence BCR-ABL gene, we find a point mutation. This point mutation is a threonine. Becomes an isoleucine in this patient. And it's threonine 315, right? Now, structurally, and we can rationalize this because this, then the left is imatinib bound to wild type ABL, and this is threonine 315. Once the threonine become an isoleucine, you can see that this isoleucine is pushing towards imatinib, right? So obviously imatinib is not bound. This is a model because imatinib is not bound onto the isoleucine 315 BCR-ABL. It cannot bind, right? Because the isoleucine is occupied part of the hydrophobic pocket that I told you about, that imatinib was designed to occupy optimally. Okay, does it make sense? So this is a resistant mutation which inactivates imatinib in patient cells. Now, this is important for a couple of reasons. Clinically, it is important because it tells us that the clinical benefits in chronic myelogenous leukemia of imatinib are most likely due to inhibition of BCR-ABL, because a single point mutation in BCR-ABL suppresses the clinical benefit. Okay, so whatever other targets imatinib is touching, they are probably not essential for the clinical benefit, right? They might be contributing to some extent, but they're not essential because a single point mutation protects you. In a one protein protects it. This is clear. This is kind of an important concept for what comes after. Okay, so, and this was a deep learned lesson for chemical biologists, because what it told us is that if I have a mutation, right, that if I have a small molecule that causes a phenotype, and I have a mutation that suppresses that phenotype, in this case, the phenotype is killing leukemia cells, right? Now I have a mutation. I can identify what mutation it is, and that mutation suppresses the phenotype, so makes this leukemia cell line resistant. And if I can confirm in vitro with the purified protein that that mutation, that compound does not bind anymore to the protein, now I have what is called the gold standard for target identification of a small molecule, okay? Which means that now that I have two different proofs that that mutation suppresses the phenotype caused by the small molecule because it suppresses binding of the small molecule. Because a mutation can do many things, right? I can mutate a pump, right? Mutate a pump, the pump gets overactivated, it pumps the compounds outside of the cells. That mutation suppresses the phenotype, but there's nothing to do with the target of the small molecule. It's a non-specific and indirect mechanism, okay? Or a mutation that makes the cell proliferate faster. That will, to some extent, suppress the phenotype of growth inhibition. It's nothing to do with what the target is, okay? But if I have a mutation that suppresses the phenotype, and I can prove in vitro that the mutation in that protein suppresses binding of the small molecule, now I have an understanding of what the small molecule does, or what are the physiologically relevant targets. Okay, so in this case, this body of studies, body of work demonstrated that BCR-ABL is the physiological target in humans of imatinib as related to chronic myelogenous leukemia. All right. Now, we can use this concept. In the last 10 minutes, I'm going to talk about this. How do we sort of leverage this knowledge now? Okay. So let's look at the graph on the right, on the left. So we have a drug, it's increasing dose. This is familiar, right? And we have a target allele. We call it allele, which is the protein wild type. Wild type means the natural form of the protein that is in the cell, in the organism. Okay. So, and this is inhibited by the compound. In increasing concentration, you have the typical dose response curve. Right now, we have a mutant target allele, This is not inhibited. Okay? Or is inhibited at a much higher dose. Okay, so you have a range of concentration in which you significantly inhibit the wild type allele, but you do not inhibit the mutant allele. Okay. Now, if I take the mutant allele that I saw in patient, for example, and I put it in a cell line that has only the wild type allele, now I should expect any phenotype that is related to my target to go away, because now those cells express the mutant protein. Does that make sense? If the phenotype does not go away, so it appears in both the mutant and the wild type at the same dose of the compound, that means the phenotype is not on target, right? It's not due to the modulation of that protein. It's due to something else. Okay? Does it make sense? So only the phenotypes that goes away in the mutant demonstrate that that phenotype is dependent on the intended target. The phenotype that stay the same in both mutant and wild type are not dependent on the intended target. Okay? So this is the concept. Now let's see how we can use this. So these are two anti-cancers, indisulam and tacisulam. They were identified in phenotypic screens. So no KD, right? So we take cancer cells and normal cells, we do parallel screens, and we ask which compounds only kills cancer cells, it doesn't kill normal cells. Simple idea, okay? Identified a few compounds, they do kill only cancer cells. This is highly simplistic because which cancer cell you use, which normal cells to use, right? A cancer cell is very much different than a normal cell. And the cancer cells are different among them, depends on the tissue, the tissue of origin, depends on the stage, the treated and untreated. But in a more simplistic way, we take cancer and normal cells, normal fibroblast and liver cancer. And we ask which compounds only kills cells that are cancerous and spares normal lung fibroblasts, okay? These compounds came out. And no knowledge about what they do, what is the target. But they were effective. They were drug-like, okay? And they were active in animals. They were cure cancer in mice. So Eli Lilly pushes these compounds through the clinic. There are about 30 clinical trials. Over 30 clinical trials were initiated with one or the other, right? Hundreds of patients they took this because it looked effective by all the standards by that time of drug discovery. All clinical trials failed. There was no clinical benefit, okay? So there's something fundamentally wrong here. And we couldn't predict anything about what particular patient population was who may get some benefit from taking this drug, right? So we lump all the cancer patients together, we give them the drug, and guess what? Statistically, no effect, okay? So now this is the idea to understand what is happening here, right? We take a cell line that is normally killed, the cancer cell line, we add a compound, or we add just the vehicle, right? So the compound will kill most cancer cell lines, but some will survive. We use a suboptimal dose, okay? We don't kill them all. So some cells will survive, and those cells will eventually repopulate the entire population, okay? So you end up with a population of cells on the left here that is resistant, and a population of cells that is still sensitive because you didn't challenge it with the compound, okay? Now we go, and these are sort of the inhibitory IC50 curves of the various isolate on that plate. You separated the various isolate, you grow them in separate culture, and then you measure the IC50s. And indeed, they are resistant, right, to different extents. Some of those isolate, they need a lot more compound to be inhibited, okay? And the black line is the wild type, the parental, we call them, the one that were not challenged, okay? Now, how do we identify mutation? What is changing in these clones that now are resistant, right? We take the entire mRNA, now we can do that, of the cells, and we sequence it, okay? We ask, and we compare it to the parental cells. We ask, what are any mutation that I can detect in these different isolates, okay? And the idea is exactly to identify mutations that may cause resistance because they are targeting the protein that the compound binds. Okay, so once we do this, these are the result. The result is the histogram on the right, right? So there are, in these 6 isolates, we have resistant, we have 551 mutations that recur once, okay? But we have only 80, too many to study, at least at that time, okay? And very, very laborious. But we have only 80 mutations that recur in 2 different isolates. Remember, we don't really know if the isolates are independent among each other, because they can come from originally from a single cell that sort of separate, right? But the concept here is, let's look for recurrent mutations, right? Genes that are mutated at the highest frequency mutated genes, okay? So only 3 genes were mutated in 3 isolates, okay? And no genes were mutated in 4, none in 5, none in 6, okay? So that tells you that what happens in the other 3, that tells you there are many other mechanisms by which we can get resistance, right? There's not a single mechanism, because there's no single mutation that appears in all isolates. But there is one that is more frequent than others. There are 3 that are more frequent than others, okay? And these 3 mutations are actually in the same protein, in the same gene, right? And they're not only in the same gene, but they're all in the same structural motifs of that protein. The protein is called RB39, and the motifs are alpha helices, right? So those mutations were all in the same RB39 cell motifs, okay? This is highly telling, right? This is very likely that this protein has something to do with how the compound acts in those cells, okay? So this exercise identified one candidate out of 50,000 genes that are normally expressed in any cell type, 25,000 coding genes, 50,000 expressed in any given cell types at any moment, right? Now we have only one candidate that we can study, okay? And guess what? We take that mutation, we put it in the parental cell line, and that mutation is sufficient. Any of those mutations are sufficient to cause resistance. We don't need anything else. We can just pop that mutation in a cell type, and we can restore normal growth in the presence of the compound, okay? Now,
Okay, so we have 3 more minutes.
3 more minutes. I think I have one more slide. Yeah, one more slide. I just want to finish this story very quickly. So we have our culprit, right? So this is a busy slide, but just let's follow through one by one. Now, this is the protein candidate, right? It's a Western blot. And what the finding was that in wild-type cells, so cells that are sensitive, I add the compound, and this protein, the candidate protein, goes away. It's degraded, right? There's no more band anymore in the presence of the compound. But when I use a cell that has a resistant mutation, nothing happens. The protein is still there. Okay, somehow the compound is causing the degradation of the protein. Okay, and I think there's something missing here. But anyhow, how does it do it? The next experiment was we precipitate RBM from the protein using an antibody, and we study what protein comes attached to it. So we need to understand what is causing the degradation, right? And then we analyze it by mass spec. So we take this precipitated with the antibodies called an IPMS experiment, and we send it to mass spec, and we identify all the protein that comes together with our protein. Also use an antibody. Okay, the proteins that were coming together with RB39 was, to high confidence, a ubiquitin ligase, which means called DCAF15, which means a protein that mediates the transfer of ubiquitin molecules to other proteins and cause them to be targeted for proteasomal degradation. Okay, so now the full picture start to get assembled, right? So RB39 must interact with this protein that we found through mass spec, which is called DCAF15, and DCAF15 is the protein that can lead to the degradation of RBM39. Okay, so in this lab, does the point mutations on RB39 makes the protein insensitive to the action of the compound, so insensitive to degradation. Okay, so the picture here, the model that was coming through is that indisulam does indeed physically interact with the RB39, right? And the mutation prevents the physical interaction. But there must be something that this interaction promotes interaction of RB39 with the ubiquitin ligase decaf. Okay, so normally the binding of indisulam to RB39 will promote the interaction with decaf15, which will then degrade the protein. Okay, that's how the compound works. And the answer, the confirmation, as usual, comes from structural biology. And many years later, that this model was proposed, it came out the crystal structures of the ternary complex between decaf15, RB39 bound into a complex, and at the interface of these two protein bound is indisulam bound that interacts with both protein at the same time. It acts as what is called nowadays a molecular glue. Okay, so it promotes the proximity between these two proteins together, binding in both at the same time, modulating the conformation so that they now are complementary, right? And increase the activity of the ubiquitin ligase, which then multiple times ubiquitinase RB39 and cause degradation. Okay, so degradation of RB39 is what kills cancer cells. Okay, so now we
have to stop it here because the students have to go to the next class. But I want to, if students have any questions, I'll just forward it to you so that we can answer any of those things. But I want to give a round of applause to our speaker. Thank you so much.
Well, thank you so much, guys.
I really appreciate it. All right then.
All
right, thank you so much then.
Okay, bye-bye.
For those of you who came later on, can you please raise your hand? Let me know your name. Thank you so much then. Take care.
Bye, Arthur. Thank you.
Bye, guys.